Okay, so in this diagram we have our export ready mRNA. Up here all along the top of the export ready mRNA we have some proteins known as CBC, the SR proteins in pink, your 5' cap. Here along the left we have our HNRNP proteins, and down here at the bottom we have our poly A binding proteins. And I know I didn't write it, but this little green molecule is supposed to be, it's known as the nuclear export receptor, and that's very important to know. And so basically now the mRNA is ready to go through the nuclear core complex, but of course there's going to be some proteins that can't go through, and these are known as nucleus restricted proteins, and these will get unbound as the RNA moves through the nuclear core complex from the nucleus to the cytosol. And just like the nucleus restricted proteins, there are some proteins that will attach after going through the nuclear core complex and go back into the nucleus. And once this is done, you'll have these other proteins like up here that are known as initiation factors for protein synthesis. And as you can see here, there's a couple examples, a couple being like this one here known as ELF4G, and the one here on the right known as ELF4E. Okay, and so now of course the mRNA has moved into the cytosol, and looks like I forgot to draw in the CBC protein. But yeah, so the initiating factors will bind to the mRNA, and this will make it ready to go over and be in translation.